Welcome to this video 6 on this course ST9 Exploratory Data Analysis, where we're talking about t-SNE, T-Distributed Stochastic Neighborhood Embedding. So we can differentiate between the different approaches that we're applying here, where we have PCA and MDS, and they use different approaches to capturing information and plotting that on a 2D plane. So PCA maximizes the number of correlated features that explain the different components. MDS tries to minimize the error between the transformed distances and the original ones. t-SNE and UMAP are different because they try to put many dimensions on a 2D space, which can be problematic in terms of interpretation. An example of this will be if we have 7,000 point sample from this circle here on the left with a small amount of Gaussian noise. We can plot how t-SNE would plot this with different types of initialization. If we use a random initialization, we can find that we get a particular pattern is more linked together. If we use PCA as a transformation for that before using PCA, t-SNE, for UMAP, the random initialization does not give us good results, and a different form of initialization can also be helpful in terms of trying to link together the coordinates more effectively. So the transformations applied for t-SNE and UMAP may not always be intuitive, and its reflection of the true underlying distances between different groups can be very skewed. So these are all unsupervised methods that we're trying, and they try to retain intragroup distances. PCA and MDS retain intergroup distances, whereas t-SNE and UMAP do not. So these are the interlabel distances here between the discrete clusters that are present, and the within label ones are the ones like D1 and 2 within um, the gray area here. So t-SNE focusing on preserving the local structure, and tries to ensure that samples similar in high dimensional space are close together in the 2D plot. It does not really preserve the global structure at all. So there's two steps for achieving this. It converts the distances into probabilities, and samples that are closer together get a high probability of being neighbors in the 2D plot, and tries to rearrange the points in the 2D plot to ensure that this is achieved, um, and match those in high dimensions as well as possible. Differences from PCA and non-metric MDS compared to t-SNE is that it can capture better nonlinear effects for complex structures. It can be good at separating tight clusters. One big thing, which is why I put in capitals, is that inter-cluster, intergroup distances are not meaningful. The axes do not have any intrinsic meaning in t-SNE nor in UMAP, and shouldn't be used for distinguishing between clusters because they are very far away or very near. The axis values have no meaning, to start that again. And what's key thing about the clues in the name is stochastic, and therefore different seeds or other parameters can mean that you get very different results in t-SNE because a lot of the initialization steps are based on random values, and therefore the same analysis can actually produce different plots at the end. Um, it's also very sensitive to the perplexity factor parameter. So what we can do is plot our data using multiple perplexity parameters. That's what we'll do in the demonstration lab. When to use it. So t-SNE is very good at looking at single cell data, flow cytometer data, and any data where you expect tight clusters to want to visualize the cluster separation more clearly. It's less appropriate when you curve out gradients or overall structure. So for example, genome sequence data, it does it very poorly because typically you have a kind of a continuous level of diversity across the different groups. There's no intrinsic clustering necessarily in genetic data. So t-SNE is really a new mapper, really inappropriate for that. In terms of the perplexity, this is the most important parameter, and this will affect the extent to which how many neighbors each point is considered as a potential neighbor. So this is kind of the balance between local structure versus global structure. So low perplexity tends to be more fragmented. Medium perplexity tends to be one that might be optimal for any new analysis, whereas high perplexity looks at more global and therefore may force clusters to merge together. Ideally, we want the level of perplexity to be less than the number of samples divided by 3. And the easiest way of navigating this is to try to do multiple perplexity values and compare the plots so that the clusters that are consistent are the ones that are present across the different plots. An example of this is using t-SNE on different data sets. So if you look at the clusters 4, 5, and 6 here in kind of beige, blue, and green, and if we set the t-SNE perplexity to be 5, these are the clusters that we get. Whereas up here, for a value of 50, we see that the clusters are a bit different from that. What we see is that if we look across 2D space and kind of draw contours on it, is that the cluster 4 moves in proximity to others, as does clusters 4, 5, and 6 as well. So it doesn't really necessarily make concrete sense in terms of trying to assign a biological meaning behind this. Another example is using UMAP, where the number of neighbors is set from 5 to 50. Obviously, the groups labeled 1, 2, and 3 in brown, blue, and purple, or pink purple. What we see is that they move across around in 2D space if we set the number of nearest neighbors to be 50. And if we look at the contour plot of them as well, we'll find that they move around a bit as well. So this means we should be cautious about interpreting t-SNE data. So then you can ask yourself some questions. What is true or false? If cluster A is far away from cluster B, does that mean that they're different? Are small clusters important for subgroups? Is the t-SNE output the same each time you run it? Does the number of points per cluster affect the original group size? And that brings us to the end of this video.